Lovely to see you all here this afternoon. And I see that we have the challenge of taking the best of San Diego for this session. You could be outside in the sun, going to the beach, or like with my son up at Legoland this afternoon, skipping school. Or you can come and see some of the best of San Diego giving incredible talks for our conference here. So it's my pleasure. I'm Dr. Eric Veary from the UCSD Department of Neurosciences and on the program committee for the conference, and I'll be chairing this afternoon's session. And our first speaker is from the UCSD Department of Biological Sciences, Dr. Gentry Patirck, and he'll be telling us about the biochemistry of memory and the conundrum of the central dogma. Dr. Patirck.
Am I on? Yeah. Well, thank you, Eric and Stuart, for inviting me. This is quite an honor to be in a crowd like this, where I've never really given a talk in such a meeting. And so it's an honor to be able to tell you about some of the work that I'm doing in my lab. I'm definitely going to keep it high level so that I can try to make, at least leave you with something that we think is really, really important for the way synapses function in the brain. And my title has changed, as you might note, but I will be talking about protein turnover, something that my lab is very, very interested in. All right, so if we, I'll start here where movie's going to play. So thinking about information about who and what we are predicated on our ability to interact with each other and our environment, our ability to recall past experiences or form new memories, our ability to learn. We know that all of this is due to the amazing communication between synapses in the brain, between neurons and networks of neurons in the brain. And in particular, I'll be talking about what's happening here at synapses. This is a video from my colleague, Terry Sanowski at the Salk Institute. Tom Bartal put this together. This is an actual reconstruction of a region of brain in rat cortex, where it's basically showing you the dynamics and intricacies of what happens in a very quick time scale, as you can see on the bottom here, where what you're seeing here in green is an axon, in yellow, a neuron, a dendrite, where it's juxtaposed to the axon in red, postsynaptic density, and where the presynaptic terminal is meeting that. As you see, calcium is influxed here through calcium channels, and then glutamate is released. A lot of things happen, of course. For instance, you'll have glutamate being taken back up by the astrocytes, as well as neurons. And eventually what happens is that AMPA receptors, or glutamate receptors on an excitatory synapse like this, will begin to flux calcium to a point where eventually this second cell that's connected to will fire in an action potential. The movie goes on where there's an asynchronous firing also, but this is just an indication of the scale of what we're thinking about and the fact that there are many, many, many molecules involved. Some of them are calcium binding proteins. Some of them are affected by calcium here in the postsynaptic terminal, as you can see. And yeah, so I'll move on from there. I also wanted to bring up a point that it's just not the signaling, the communication that takes place between neurons. We can actually learn something from our prokaryotic friends. And this is a paper that came out last month from my colleague in biology, Gurul Sewell, where he was actually showing that there's electrical coupling allowing for nutrient time sensing between biofilms and bacteria. So this is very, what you're looking at here is membrane potential through thioflavin T, and these biofilms are literally synchronizing their growth rate through a potassium channel electrical cell-to-cell signaling. So I thought that's very interesting to even consider that prokaryotic organisms have to somehow navigate their environment and communicate with each other. So the major focus of my lab is the role of protein turnover and synaptic plasticity in neurodegenerative disease. And in particular, we're interested in the regulation of protein homeostasis, signaling dynamics, and function. As you might imagine, a lot of the work that we're interested in is happening here at the business end of the communication between two neurons here at the synapse. And one of the rules that we understand to be true is that modulations in synaptic strength, otherwise known as synaptic plasticity, are accompanied by changes in the protein composition of synapses. And a great example of why that's important is, for instance, here, if you're recording from the CA1 region of the hippocampal acute slice, and you're stimulating the CA4 collaterals and recording here in the CA1 region, this would be your test stimulation before our stimulation, for instance, high-frequency stimulation or low-frequency stimulation. What happens is that you can get a potentiation or depression of that synapse depending on what type of stimulation that you do. And what's known is that, for instance, in part, it's the change in the amount of neurotransmitter release or the change in the number of the receptors that are on the postsynaptic membrane. But something that my lab is very interested in is the contribution that making new proteins or degrading them, how that contributes to these modulations in synaptic strength. And so that's what I'll be telling you about today, mostly in a very high level, just trying to get the main points across with a few examples of data. The reason we know that protein synthesis and protein degradation are really, really important because, for instance, if you take inhibitors of synthesis or inhibitors of some of the major protein turnover pathways inside of cells, you pretty much block most forms of plasticity or alter them in some manner, as well as behavior. And another thing that we consider is that since, neurons are excitable cells, and even though a large majority of the work on protein synthesis and protein turnover and the pathways that I'll be talking about today have been studied in non-excitable cells, we have to operate here in excitable cells. So the prediction would be that the balance of synthesis and degradation would be itself regulated by synaptic plasticity. And in fact, that's one of the major things that I'll be pointing out to you today, is that these core pathways inside of cells for making new proteins, whether that be locally at synapses or degrading them, is themselves regulated by neuronal activity. Okay, and as an example, even to describe the idea that there may be synapse specificity, this is just a model just showing you that perhaps when you have an activated or a controlled synapse, you might actively degrade that a said protein at a synapse or stabilize it. And again, this is just a model of thinking about active degradation or passively stabilizing or degrading everything else in a dendrite. This idea about the central dogma, right? So I mentioned that in my first slide in my title, where we know that DNA produces RNA. DNA directs the synthesis of RNA molecules, while RNA directs the synthesis of protein. DNA being the blueprint, but RNA and protein do not live forever. And so something that I'll come to towards the end is this conundrum of the fact that we have stable memories for years, yet we are constantly turning over our proteins. What do I think about with regards to protein turnover? It's a combination of the synthesis and degradation of proteins, where half-life of a protein is required, is a required time to produce half of the protein in the cell decreased by half in the cell. And so some other general considerations that I think about with regards to protein turnover is that obviously an inherent rate of turnover for a particular protein would be a combination of its synthesis and degradation. That cell biological signals are capable of altering rates of synthesis, degradation, or both. And in neurons, this could be quite important when we think about the dynamic nature of overall protein stoichiometry of synapses in functional relevant microdomains such as synapses. The other thing is that, as I mentioned, protein synthesis and protein degradation can be themselves regulated by neuronal activity, and I'll share with you some examples of why and how that works. Why do we care about protein, balancing protein synthesis and protein degradation, especially in excitable cells and cells like neurons that are fully differentiated and not dividing? Is that it's really important that you balance synthesis and degradation for normal cellular health, because what happens in a lot of neurological disorders or neurodegenerative diseases, you get an imbalance in this, and this could wreak havoc on the cells, causing cellular death, or in some cases, altered synaptic function, and something that we think about a lot in my lab. This is a pathway that I studied, and this comes from the original title. It just so happens that the ubiquitin pathway has been studied by literally hundreds of investigators across the world, and it is the major pathway for turnover in eukaryotic cells, where various proteins can be modified by a small protein called ubiquitin. It's a 76 amino acid protein that's conserved from yeast to man, and it's really this tagging of other proteins that sends them to a particular fate. For instance, one of the things that we study is their degradation at the proteasome, and I'll share what that is in just a second, or other cell biological events such as promoting the endocytosis of membrane proteins and their endocytic sorting to, say, a recycling or a degradation pathway, as well as a whole host of other things that the tagging of proteins by this pathway can happen. There are actually literally hundreds of these enzymes that covalently attach ubiquitin to other proteins. They're called ubiquitin ligases here in the E3s. And it's known that, a large majority of your proteins are undergoing protein degradation through this pathway at any given time. In fact, it's about 20% that can be modified if you were to take a snapshot. So what happens to proteins that are modified by ubiquitin? Well, one of the things is that they can be degraded by the proteasome, shown here on the left. Additionally, something that my lab is very interested in is the trafficking of membrane proteins to an endocytic fate, where you have to either decide to be recycled or to be turned over at the lysosome. And so I'll tell you a bit about what we've learned thus far. For instance, one of the things we know, as I mentioned just a second ago, is that a large number of the proteins found both on the presynaptic and postsynaptic terminal are modified by ubiquitin. And again, we found that about 20% of the synaptic proteome is modified by ubiquitin. This is a schematic of the proteasome, which is this large multi-subunit proteolytic complex, which has a variety of activities. It has an alpha and beta subunit proteolytic activity in the core, 20S core, and this cap here, the top and the bottom, is a 19S regulatory particle that actually identifies ubiquitin, pulls the protein into the core of this machine, unfolds it using ATP, and literally chops it into pieces. And this is how all MHC1 Cas peptides are made, or the large majority of the turnover that you see in eukaryotic cells are going through this pathway. And as I mentioned, we care about how this pathway might be regulated by neuronal activity, and this is just an example actually from my previous advisor, where they showed that if you were to tag, say, put green fluorescent protein, GFP, on one of the subunits of this multi-subunit proteolytic complex, and then ask whether or not, if you stimulate neurons, what happens to the distribution of this complex. And what they found was that it rapidly traffics into dendritic spines, suggesting that this trash can cleanup system, the degradation pathway itself is receiving instructive cues by synaptic activity. And over the years, my lab and a few other labs have contributed to an understanding which this model here, in that calcium through the NMDA receptor activates CAMK2, which then interacts with this proteolytic complex and literally sequesters it into dendritic spines at synapses, and then you get a cascade of protein degradation. Of course, we'd like to know all the proteins that are turned over, and I'll give some consideration to some of the big questions with regards to protein turnover in neurons later on. This appears to be really important because we found, for instance, that when we made mouse models of this system, where we created phosphomimetic and dead mutants of the actual site of phosphorylation on one of the subunits that was targeted by CAMK2, there was a variety of things that we uncovered. For instance, spine dynamics seem to be altered. It appears in a collaboration with Karen Zito at UC Davis that proteasomes are actually required to make new spines in an activity-dependent manner, suggesting that, and I maybe should have mentioned this earlier, whenever I tell people that I study synaptic plasticity and that I study protein turnover, a lot of people think of the fact that, oh, maybe you're degrading memories or degrading synapses. But in fact, you need turnover actually to not only weaken synapses, but you need them to strengthen synapses, because you're getting rid of, for instance, proteins that might be restricting that plasticity event. Okay, so this was a large part of my lab has been working on regulation of proteasome trafficking and sensing these synaptic cues. If we come back to this model here, again, and I'll be giving you a couple examples of where neuronal activity is literally regulating the pathway. The other half of my lab is very interested in understanding the trafficking of AMPA-type glutamate receptors, which I mentioned. One way that you can downregulate or degrade various membrane proteins is their tagging by ubiquitin. And it turns out that AMPA receptors are actually modified by ubiquitin. My lab discovered that, and this modification leads to their degradation. Here's another schematic of what this looks like. I'm sure many of you know about glutamate receptors, but AMPA receptors mediate the large majority of fast-acting synaptic transmission at excitatory synapses. And they're needed to deliver a local depolarization so that you can activate NMDA receptors, which are your coincidence detectors, detecting both glutamate binding and membrane depolarization. A lot of things happen. For instance, calcium comes in, and then you can activate and control a lot of signaling pathways. What that means is that there are literally hundreds of scientists who study the trafficking of AMPA receptors, meaning the trafficking of AMPA receptors to and from the synapses. And for instance, in early days, it was understood and identified that there were silent synapses that only contained NMDA receptors. And so the recruitment of AMPA receptors was a way to activate them. And then you had this dynamic population of silent synapses and active synapses. And developmental and use-dependent regulation was something that was very critical here. There are rapid phases of plasticity involving the rapid internalization of these receptors or exocytosis of these receptors, and that yields a depression or potentiation of that synapse respectively. So indeed, you can almost think of the number of these receptors on the synapse as a correlate of the synaptic strength of that synapse. There's another form of plasticity called homeostatic plasticity or scaling, which is basically a compensatory mechanism to deal with chronic activity changes over periods of days, hours to days, so that if you upregulate activity for a period of days, you will then have this compensatory mechanism to decrease these receptors. Or if you inactivate synapses, say with tetrodotoxin, which blocks all sodium-based action potentials, you'll start to slam more of these glutamate receptors into the synaptic membrane. So what my lab did, we discovered that there is a ligase that responds to neuronal activity and ubiquitinates AMPA receptors. And this was a very old model where you see these receptors modified by ubiquitin and then eventually making their way to the lysosome to be degraded. More recently, we've uncovered, and it was very nice to find a ligase that had a C2 domain. A C2 domain is a calcium and lipid interaction domain. And it made sense for us to think about this in the context of activity dependence, in that perhaps this ligase might itself be regulated by neuronal activity. And what you're looking at on the left is expression of this ligase under basal conditions. And within minutes after stimulation, we literally traffic all of this ligase to the synapse. And this has been a continuous theme in the research that we've been doing, indicating that you kind of keep all of the degradation machinery out of the synapse until you need it. We're understanding mechanistically how this is taking place. We also know that in this model here that this ligase appears to be working in downscaling, or this compensatory mechanism to upregulation of activity. What we were able to do was to take bicuculin, which is a drug which blocks GABAergic synaptic transmission, thereby having enhanced synaptic transmission. If you were to apply this to slices or to cultured neurons, I did fail to mention what model we work in. We work in rodent, and we work in mainly organotypic slice and cultured neurons, and we do work with animal models when we've translated some of this stuff for behavioral analysis. Anyway, what we found was that the application of bicuculin, which enhances synaptic activity, literally takes a pathway that now drives these receptors for degradation, utilizing this ligase. So if we were to use a hairpin to knock down NET4-1, this ligase that we've identified, it would prevent the bicuculin-induced downscaling that we see in neurons. This other enzyme called USP8 is actually counteracting this. And interestingly enough, what we see not only in downscaling, but in some disease scenarios where NET4-1 levels are increasing while this thing that's counteracting its function is decreasing. This is from a review, or actually, no, this is from a recent paper from Aaron Schumann, my postdoctoral advisor, who's now was at Caltech when I was there, but now at Max Planck in Frankfurt, where she did a similar experiment looking at the number of proteins that were increased or decreased in response to upscaling or downscaling. Okay, so the way that works, if you throw on TTX for some period of time, you're upscaling because you're silencing the neurons. If you throw on bicuculin, you're downscaling because you're upregulating this. And what they found was that the number of proteins that increased or decreased did not change between any condition, but it was actually the up or downscaling caused an opposing translational regulation of several molecular pathways. So it appeared that certain pathways in total, in cohorts, were bidirectionally regulated, which is allowing to produce, an adjustment to the proteome that can be fine-tuned. And this is looking at synthesis. We always think about an aspect of our work with relation to disease, because most neurodegenerative diseases, if you've never thought about it before, are actually diseases of altered protein homeostasis. And that goes for Alzheimer's disease, Parkinson's disease, Huntington's disease. These are all diseases of, at some level, diseases of altered protein homeostasis. So we asked a very simple question about this ligase, whether or not it was involved in A-beta-induced synaptic depression. And of course, many of you have heard of Alzheimer's disease, and A-beta being the cleavage product of the APP protein that is at play in producing a lot of the toxic or effects on synapses. And what we found was that this ligase is also being trafficked to synapses in response to A-beta. suggesting that there must be similar mechanisms where this pathogenic signal, as well as normal synaptic activity, is converging or utilizing the same molecular mechanism to bring this ligase to the synapse to cause the receptor to cause the ligase to ubiquitinate the receptor and downregulate it. All right, so back to this model here, and that is we started to think about one other component, and again, this is a theme, right? This machinery that we find in neurons to be regulated themselves by activity. We begin to look at lysosomes for a particular reason, and that was that it was thought that lysosomes are mainly in cell bodies. They can traffic in axons, but it's thought that dendrites are devoid of lysosomes. And what is a lysosome? It's, if you know your basic cell biology, it's a membrane-bound organelle that has a low pH environment and a lot of hydrolytic enzymes in it. This was a large-scale collaboration between myself, Brenda Bloodgood's lab, and Mark Ellisman's lab. And we asked a very simple question because not being a bona fide card-carrying lysosome cell biologist, I can naively ask, well, just because you don't see them there, maybe it's a possibility that, you know, functionally they're there and you just haven't figured out, or maybe you haven't looked hard enough, or actually there may be another way to characterize whether or not they're functioning there. And the question really is, you know, membrane proteins out in distal dendrites, how are they going to be degraded? You're either going to bring lysosomes there, you're going to bring the cargo back to the cell body, or they're going to maybe be these outposts. The middle model seems pretty precarious for things that might aggregate and basically interrupt trafficking and function of dendrites. So we simply asked, where are lysosomes in neurons? And it turns out that using a variety of methods to actually prove, involving EM and functional assays, but just to give you an indication that they are out in distal dendrites. They're mainly in the cell body, but you find them in the base of spines, you find them in spine heads. There's a heterogeneity in size. Here's a 2-photon image of one in a spine head. And not many are actually found in spine heads, but there's about 6 to 7% that we find in the dendritic spine head. And so one of the things that we wanted to ask, I'll skip over all the data that went within this body of work that is just, we just were able to convince the reviewers that it was worthy to be published. And what we did was to take organotypic slice culture from rat, from the hippocampus. We could biologically infect these or transfect these neurons with this LAMP1 GFP construct, and then basically image them using 2-photon imaging. And what we wanted to do actually was to ask if we uncage this idea that neuronal activity might be regulating the function or distribution or trafficking of this organelle, we asked, if you uncage glutamate at a single spine, what might happen to the distribution or the function of the lysosome? And what we found very strikingly was that after stimulation using uncaging over a spine, that more than 90% of the stimulated spines recruited a lysosome to the base of that spine, while only 41% mock stimulated recruited a spine. And the duration of that lysosome organelle was actually at the base of that spine was much longer. And here's a kind of a higher resolution look at this, where you have lysosomes actually traveling along microtubules. What's not interesting is that they stay along their tracks. They don't really jump tracks. That was very interesting for us to see. Here's one that stopped here, and we're going to uncage glutamate right at this spine, and we find that they actually get captured at the base of a spine, suggesting that indeed there must be instructive cues whereby the lysosome organelle itself is receiving. Okay, so the large majority, giving you mainly kind of highlight experiments from some of the work that we've been doing, but indeed what we've been really interested in is understanding how neuronal activity is engaging these pathways to control the fate of turnover of many proteins. There's a lot of future perspectives that we want to think about and future studies. For instance, the turnover of which proteins, are required for learning and memory. How is the synthesis and degradation of cohorts of proteins co-regulated by synaptic activity? What controls the timing and location of synthesis and degradation at synapses? And how do these mechanisms couple to classical signaling for synaptic plasticity? And lastly, how is protein half-life altered during learning-related synaptic plasticity? Okay, and so we're engaged in a lot of experiments that are addressing some of these questions. This field is actually very young, in that we know very little about the proteome at synapses with regards to what's being made, what's being turned over, how long do they live. But these are some of the things that we're starting to address. I want to leave you with back to this conundrum idea, right? So the biochemistry of memory addressing the conundrum of the central dogma. And I already mentioned that the central dogma is a scenario where proteins don't live forever, but your memories can be long-lived. So, and as I mentioned, I showed you guys this. So let me give you a quote from one of my colleagues, actually, Terry Sanowski, who's at the Salk and also in my department at UCSD. In 2005, I believe, he proposed the following: I have been puzzled by my abilities to remember my childhood, even though most of my molecules in my body today are not the same as the ones I had as a child. Despite this molecular turnover, I have detailed memories of places where I lived 50 years ago, memories that I never rehearsed but which are easily verified. If memories are stored as changes to molecules inside the brain, molecules that are constantly being replaced, how can a memory remain stable over 50 years? And it just so happens that this is a very interesting idea, and he proposed a model. And indeed, what we found later on was that another colleague at UCSD who recently passed, Roger Chen, received the Nobel Prize for GFP. He proposed that long-lived memories might be stored in the pattern of holes in the perineuronal net. And that's basically this extracellular matrix around neurons that are made of a variety of proteins cross-linking within the extracellular matrix. So it's interesting that perhaps while proteins inside of your cells are turning over, some of the most stable proteins that are actually found are actually out in the extracellular matrix. And what Roger proposed was that these holes within the perinuclear net would be these stable tracks for where that would restrict synapse, the dynamics of synapse loss and gains. And so it's interesting to think about the extracellular matrix as maybe one potential place where you might be able to permanently store memories. Interestingly enough, part of the reasoning was that metalloproteases appeared to be involved. And just recently, there was a paper that came out a few months ago in Neuron, where they showed that the activity-dependent exocytosis of lysosomes, something that I really never had a consideration for, was that lysosomes are constantly fusing with the plasma membrane. And it turns out that they're delivering activated cathepsins. And these cathepsins are activating these metalloproteases, and they then go on. In this case, they showed that they go on to activate these metalloproteases like MMP9, which is important for sculpting and modifying dendritic spines. and that if you blocked the exocytosis of lysosomes, that you would alter spine dynamics. So I'll leave you with that, and perhaps you'll think more about protein turnover at synapses, the idea that you have to maintain this balance between synthesis and degradation, and somehow that all has to be controlled in a way that allows for these stable memories to be maintained. So I'll stop there.
Thank you very much. Thank you. You show this, your model is the spines, and you show the local microdomain, and you have this incredible animation of the lysozymes going along the microtubules. So is it possible that internally to the axon are control or computational elements that are making all this happen?
Yeah, we don't know. Well, we've just now started to look at the dynamics of this trafficking. We believe that there's likely some regulation of motor proteins on microtubules. I think that's what you're getting at with regards to, what's allowing them to actually be recruited to synapses. One of the things we understand that we found in this body of work that they seem to be tethered to F-actin, and that when we actually disrupt F-actin with latrunculin A, we see that they move faster. Of course, when you knock out microtubules with nicotazole, they stop completely. And there's also a very interesting aspect of dysfunctional lysosomes getting larger and having an issue with motility.
Questions? Down here?
A very beautiful presentation.
Thank you.
I wanted to ask you, however, there's a rich literature about messenger RNA stored locally to the spines. Have you encountered any of that in your research?
I had one slide that was a nod to that. In fact, I did my postdoc with Aaron Schumann, who's one of the premier persons who studies local protein synthesis at synapses. And when I got to our lab, I said, do you want to study local protein degradation? Yes, I had not been looking at synthesis, but I know that it's a consideration that we have to have because of this balance that needs to be maintained. And yeah, it's interesting that you have mRNAs, not all mRNAs, certain mRNAs being trafficked by ribosomal on RNA binding proteins down microtubules to certain synapses.
Yes, yes. And in a similar vein, I had a second comment. You mentioned the extracellular matrix. Have you also considered the subsynaptic matrix, where the actin filaments are going to be interacting with the microtubule filaments and guiding transport and so forth?
No, not until minimally now, where I'm thinking about organelles moving along, say, the microtubules. But it's interesting to think about F-actin, right, as this right underneath the mid, and whether or not lysosomes, proteasomes, things that are degrading, or things that are being made that are involved in allowing new spines to be formed. I don't know if you guys know, but spines are always being made and they're always being removed. They're not stable. There are some stable ones, but there's a lot of dynamics, and so that could be involved.
Yes. Well, thank you. Thank you very much.
No problem. Thank you.
Down over here.
I see your hand. Yeah.
We'll go there and then there and then there. Go ahead. Go ahead. Yeah.
Okay, so I have a question. Can you hear me?
Go ahead.
Yeah.
When you're talking about the protein turnover, and coming back to the question when you say, how come proteins are turned over all the time, but memories persist per years? Do you really mean that all AMPA receptors are reuptaken in the synapse and the new synapse is being formed, or just the receptors are being recycled, but the synapse remains?
So AMPA receptors, they have a finite half-life, some of them on the order of hours. We actually don't know what the half-life of receptors are under various plasticity paradigms. We have basal control levels using S35 at a minimum. It just so happens that 80% of AMPA receptors are internal, only 20% are on the surface. And those on the surface, they're not there forever, right? They are constantly being internalized, and some of them are recycling back. But under certain plasticity paradigms, there's a shift where, indeed, some of them are being then degraded. We just don't know all the details yet to make a clear sense of, how long they're staying under certain parameters or not. But they don't live forever.
Right. So my question is the new synapse formed? Are they actually disappear and the axon is able to make a new connection on the new dendrite? Because otherwise the memory still remains within a circuit. As long as the synapse is maintained, it doesn't matter the receptors are recycled.
Right. So as long as you have a synapse there, you will be bringing new receptors there. I think is your question, if you remove all receptors, will the synapse disappear? Like what comes first, chicken or egg type scenario?
Yes, in the terms of maintaining the memory, like you're saying, why do you even assume that with the protein reuptake and the protein recycling has to do anything with the memory, and it's not just maintaining the synapse?
Right. Well, I would say the last part of my talk is a divergence and a disconnect, something that I don't really think about that often. The large majority of the efforts in my lab are really understanding why you have to have protein degradation as a rate-limiting step for plasticity changes. Now, this idea that proteins don't live forever and synapses aren't there forever, but however you still maintain these memories, that was to be provocative at a minimum. And also to actually think about, it is in the context of making proteins and degrading them. And maybe that model at the end is not right. Maybe it is that you're replacing proteins in a way that you're maintaining the synapse.
Right. Just the last thing I'll say, the metaphor that I thought of is like building a bridge. A lot of people come to build a bridge, and new people come to build a bridge, but the bridge remains there. It's been built.
Yep.
It would be more interesting to see how is the memory maintained when the neuroplasticity is happening and new synapses are formed, but memory still remains. Like when the circuitry changes.
Yep.
Thank you so much.
One last quick question.
Since this is a real university type question, I'll say Norm Kachuk, USC.
Okay.
Do you think that there's a cross-species preservation of all this mechanism so that it looks the same in precordates, chordates, et cetera? But also, since we've not talked about epigenomic changes that can alter the synthesis degradation cycle of a lot of this, whether or not we're going to see more complex and different issues in human brain, even, let's say, other brains that don't have the neuroplasticity that we see, at least phenotypically.
Right. So is this, where is this stopping along the evolutionary chain? We've only looked in, well, that's not true. We do know that these pathways are acting, for instance, even in C. elegans. So the glutamate receptor that Josh Kaplan at MGH studies is that he found that they are modified by ubiquitin also, and so it appears that this pathway may be working even in C. elegans. Whether or not other things like epigenetics or control is going to be something that's more specialized or important in this, I would say that we haven't looked to understand whether or not the epigenetics is another layer controlling the balance of synthesis and degradation, but I would say that indeed we just haven't looked, and it's probably the case, right?
Let's do it.
Yeah.
Okay. So yeah, we're well out of time. Thank you.
All right,
thank you so much, Dr. Patirck. And now carrying on with the stellar pattern of scientific discussion from UCSD, we have the illustrious Dr. Ramachandran talking about embodied brains and disembodied minds. Dr. Ramachandran.
It's wonderful to be here at this conference. Can everybody hear me? Thank you. I'd like to thank Stuart Hameroff for inviting me to present some of my ideas here. And he is to be congratulated for creating this whole conference and making it possible. Now, when thinking about consciousness, there are two problems which are confounded, I think. That's my view, it may be wrong. One is the metaphysical, philosophical problem, which I call the vantage point problem. It comes from the Upanishads in ancient Indian philosophy, revived by Erwin Schrödinger. And the other one is the empirical problem of how the brain constructs a sense of self. And I'm going to describe each of these in turn. The metaphysical problem is the following. Physics does not acknowledge the existence of first-person singular. In physics, the I does not exist. There's only an observer, and interacting with the universe. There are billions of brains, thousands of brains, of which one is mine. My brain has no privileged access to the universe. And yet, from my point of view, the I is everything. I'm looking at the world from inside this skull. And this paradox, this asymmetry in nature between the third-person description, the objective description of physics, and the internal subjective viewpoint, I'm looking through this window at the world. That asymmetry is a metaphysical problem, and I think it won't be solved till we make advances in physics and come up with a new physics which can incorporate that vantage point. And this is a point made by Penrose and others. Now, I'm not going to talk about that, the metaphysical problem of vantage point, first-person singular, qualia. Qualia also arises only from my point of view. In physics, there's no qualia. So unless you change physics, you can't explain qualia. As simple as that. Now, coming to the empirical problem, many ways of tackling it. One way is to look at patients. And this is what we've been doing. So that's what I just said. Okay, patients. Or even just introspection about your own self. What are the various attributes? The most obvious attribute is recursive self-awareness. You know you are conscious. You have a sense of self. The most obvious and yet most paradoxical and elusive. Second, there is the embodiment of self, the notion that you are anchored in this body and not anchored in Stuart's body. Again, seems axiomatic, but it's quite subtle. And you can change, using a bit of optics, you can change that. You can be in somebody else's body. Third, there's a sense of unity, the fact that you're one person despite a staggering diversity of sense impressions, memories of a lifetime. Your sense of continuity in time is another one. Sense of agency, free will, that I'm in charge of my destiny, or even picking up a toothpaste from the desk in front of me. Then there's a link with language I won't go into. So there are all these many different attributes, and I believe you can study these empirically in the lab, and then ask yourself, how does the coherence come about? You have the sense of one person engaging in all this activity. So let's first talk about embodiment. And the way we stumbled on this is, well, let me give you a few examples from neurology which are less well known, but I'll get to that later. Let's first get to embodiment. You all know about phantom limbs. Arm is amputated, leg is amputated. You continue to vividly feel the presence of the phantom. Widely publicized, so I'm not going to talk about that except in passing as we go along. I'm going to say a little bit about the exact converse of this, but a person who's completely normal. I met a dean from an engineering school the other day, and I won't mention his name, obviously, and it was not an engineering school, somewhere else, who wanted his arm removed. He came to my office and he said, he retired at the age of 70, and he said, all my life I harbored this urge to have my arm amputated. I never told anybody, and I'm just coming out. And he took a felt pen and he drew a line along which he wanted the arm removed. And I had heard of this phenomenon the first time I was seeing it, and I went and did some research on the internet, and there are chat groups. There are groups of people who want to have their arm or leg amputated. And he was a perfectly sensible guy, and I had a long chat with him, and I said, you sure you want to do it? And it's only legal in Mexico and in Canada. It's not legal to amputate a healthy limb in the United States. What causes this phenomenon? So the first thing I asked him is, what do you feel like it's alien or something? Why do you want it removed? He said, no, it's not alien. On the contrary, it's intrusive. It feels like it over belongs to me. And when you're a physician or a scientist studying neurological patients, one of the first things you learn is listen to what he's saying, right? He's saying it actually over belongs to me, it's intrusive. What does that mean, right? So we first said, maybe if you look at the map in S1, that blue strip there is not actually blue, of course. That blue strip with where the somatosensory cortex is, the Penfield map, but there's a complete map of the body surface and the surface of the brain. Behind it is S2, which is a map of proprioception, joint, muscle sense, and all of that. Maybe that map is congenitally missing the arm, because this guy says ever from childhood, he had nurtured this desire to get rid of the arm. Goes that far back into childhood. Some of these people go on, about a third of them go and get the arm removed, by the way. They get it removed, so it's a serious clinical medical problem, and they get it removed, and they feel happy. They're perfectly, usually 90% of the time they're happy, and the depression lifts, and they're very happy about this. But what's going on in the brain? Well, people looked at this, and people have known about this for a few decades, and the Freudians, some psychoanalytical folk, have described it as a wish fulfillment. The guy wants to draw attention to himself, so he wanted the arm removed. And this doesn't make any sense to me. I mean, why not remove a ear or something innocuous like that? Something as drastic as removing an arm for engaging in wish fulfillment doesn't seem sound right? The other claim, which is even more absurd, which I found bizarre, is that you amputate your arm to create a stump which resembles a giant penis. And this is found in the textbooks. If you don't believe me, you can go check it up, right? Again, this didn't make any sense to me. I said, maybe there's an anatomical, physiological basis for this. So I said, let's go and look at the Penfield map, which is there in coronal section, and you all know about the Penfield map, the disposition of the map. Oh, I can't, this thing, I can only show it on one. I'm sorry, the spot's not very visible, but it's a person draped across the somatosensory cortex. Systematic map, except the face is near the hand instead of being near the neck where it should be. And something funny here, the face is right side up, the rest of the body is upside down, so the upper part of the face is near the hand. So anyway, other than that, it's a continuous map. And I said, maybe the arm is missing in that map, congenitally, for some reason. I mean, therefore, he finds the arm to be alien, okay? So we mapped out S1 using MEG, and it was completely normal. Then we said, okay, well, there goes that one, right? Then we measured his galvanic skin response by poking different parts of his body and seeing if you get an aversion response, you get a skin response. Normally, if you take any one of you and I poke you with a needle, message goes to the somatosensory cortex, to the insula, thalamus, insula, anterior cingulate, hypothalamic nuclei, and then makes you sweat. Now, if that pathway is messed up, maybe he won't sweat if you poke him in the arm he wants removed, because it doesn't feel like it's part of him, right? So I then poked him below the line of amputation. We want once the amputation done, and above the line, normal hand, neck, chest. He gets a normal GSR everywhere. Below the line of amputation, I thought he would get a decreased GSR. In fact, it went through the roof. It was higher, much higher. So I said, there goes that one again. My God, and he's getting, but it fits with what he's saying. It's overrepresented, right? So he said, what's going on here? But what we found was Paul McGeer, and he's a postdoctoral fellow in our lab, and mainly Paul McGeer and David Brang, who was a graduate student at that time. What we found was that if you go to the next area higher up, you see the S1 and S2 project to IPL, but also to SPL, superior parietal lobule, in the parietal lobe, which constructs a body image. You know, close your eyes, you have a vivid sense of your body, using proprioception, muscle sense, joint sense, vision when you open your eyes, verifying your hand is there, hearing, vestibular sense, all of that converges to create a body image. That map is missing the arm. So what's going on here is the messages go from the hand, the skin is normal, everything is normal, S1 is normal, sensory signals are processed normally, goes to S1. From that, the message is delivered to the superior parietal lobule, body image. There's nowhere for it to go to. So there's an acute discrepancy. Discrepancy kicks in, and God knows where that's picked up. It goes probably to the amygdala. And then it's picked up, and it's felt as acute anxiety and a desire to remove the arm. Remove the arm, the patient's fine. So this is an example of how you can take a bizarre, incomprehensible syndrome and then reject the Freudian view and come up with a reasonable explanation in terms of neuroanatomy. Okay, now that's about apotemnophilia. Let me now switch gears, go back to phantom limbs. Now we said it's been known for 1,000 years, but let's say maybe we can do some experiments on them instead of just presenting them at grand rounds and saying the patient is a phantom limb. Can we do any experiments? One of the earliest experiments we did was to simply do sensory testing, and we found that during routine neurological testing, we found something quite strange. Although we had actually expected this, we predicted this, so we went to look for it. We found that if you touch different parts of his body with a Q-tip, with his eyes blindfolded, that's my nose, doctor. That's my chin, sir. And you remove, his arm has been removed from just below the elbow, or in this case, above the elbow. He's got a phantom left arm. You touch different parts of his body. That's my chest, sir. That's my shoulder, sir. He reports it accurately. Then you touch him here, and he says, oh my God, you're touching my phantom thumb. That's my phantom index finger. That's my phantom pinky. That's my phantom ball of my thumb. There's a complete map, a topographically organized map of the missing phantom hand on the surface of his face. And we mapped it out and then sent him home. They're not high-tech science. How do you know he's not making it up? But why should he make it up? He doesn't know anything about Penfield or maps or anything like that. But granted, we have to prove it. So the guy went home. Unexpectedly, suddenly we brought him back and then remapped it. It was the same map. It's unlikely he had memorized it and drawn it when he went home. Okay? And the map is always roughly in that region, lower face, that region, clearly delineated receptor fields. And on a whim, I said, well, first of all, why does this happen? Well, let's go to the Penfield map. That's the Penfield map. So when the arm is removed, and there are strong hints from animal literature, Mergenek, John Cass, and others, showing you get this massive reorganization of the cortex when you do a deafferentation. Here you have an amputation, which is a drastic deafferentation. Sensory input to the cortex has been removed. So the sensory input from the face skin, which normally innervates the face, invades the vacated territory corresponding to the missing hand and activates the cells. So there's a cross-activation. So the sensory input from the face skin goes to the face area normally, but in this case, it invades the vacated territory corresponding to the missing hand, activating the hand cortex. Whatever is reading the signals higher up says that's my missing hand. So when you touch his face, you feel it in his hand, precisely as you predict from the Penfield map. Now, that's fine, and then we said, let's have some fun. Let's put a block of ice on his face. What happens then? The ice water started, then he said, I feel cold. My finger feels cold. My thumb feels cold. My pinky feels cold, depending on where you put the ice cube. I put hot water, Q-tip in hot water, and then put hot water on his face. Said, my cheek feels hot, but my thumb also feels hot. Pinky feels hot. This is very strange, sir. And then, so the reorganization that's occurred is modality specific. For temperature, warmth, cold, it's not higgledy-piggledy. For vibration, you put a vibrator on his jaw, he'll say, my thumb is vibrating. Now, the interesting thing is, one day the water trickled down his face accidentally. And he said, oh my God, that's strange. I said, what? The water is trickling down my phantom. And he took his normal hand, followed the path of the trickle down to his wrist. And just for fun, I said, raise your phantom, point to the ceiling. Raise your stump, point to the ceiling. Put the water again, started trickling. He said, my God, the water is trickling uphill, defying the laws of gravity. And we've seen this time and again in patients. They don't make it up. It's because of the precision with which the topographic reorganization has occurred in the cortex. We can verify this using MEG or FMR. We've done that. But before I go there, there's something odd here. I've been misleading you slightly, as though all the pieces fit. Now, there's something wrong with my explanation in that I said there's a remapping, sensory input from the face skin, invades the adjacent territory in the brain, the hand area, right? And therefore, when you touch the face, he's experiencing sensation in his hand. Fine. But you look at the diagram and think of the Penfield map. There's something wrong with what I just said. Anybody here? I'll put you out of your misery. The sensations are always from the lower face. We've never seen something coming from the above, very rarely, always from the lower face. Now look at the Penfield map. What's near the hand? Upper head, not the lower face. So what's wrong here? So I said either, this is right side up, the face is right side up, so I said, either Penfield's wrong or I'm wrong. And I said, obviously he's wrong, he's a surgeon. Okay, and I went and looked at his data, and I found it's based on very few data points, average across dozens of subjects. So even though overall disposition of the map is correct, we give him credit for that, he got the head upside down. And I said this in a lecture in Stanford, and a young resident there, he said he was doing FMR on the Penfield map right then, that week. He went and did it again, and he said, he wrote back to me, emailed me, said, you're right, the head is, he got the head wrong, you got it right. And I'm very fond of this, not because it's very great theoretical importance, but because the first time, I think it's a good feeling, you can go and play around with a Q-tip with the patient and correct the neuroanatomy. So they could change all the textbook diagrams from now on, okay? All right, so that's that. Now let's go on to, okay, here's the I'm just showing you the corrected diagram. MEG. Now, how do you treat phantom pain? And let's look at that question. So patients with phantom limbs often say they can move the phantom, and that's that. But about a third of them will say, my phantom, it does not move, it's rock solid, it won't budge. I try to move it because it's excruciatingly painful position. He'll mimic the position with a normal hand, an awkward, hyperextended, so that the dorsum of the hand goes backwards and touches the wrist, which is impossible in real life. In movies you see it, but in real life you can't do it. But the patient's hand does that. And it's excruciatingly painful when he does that. Or it goes into a clenching spasm with the nails digging into the palm. Or it's an awkward position, it's very painful, and it's a serious clinical problem. They get depressed, often, sometimes contemplate suicide. So we said, what's going on here? I'm going to not go into a lot of detail on why we thought of this, but we thought of a simple procedure to revive make the pain go away. So I said, is it the main reason of the pain, an awkward position? He said, yeah. And we said, why don't you move it? He said, I can't move it, it doesn't move. When I looked at the case sheets, one of the things I found was these people often had a pre-existing nerve paralysis, brachial avulsion or some paralysis. Arm was lying in a sling in an awkward position, paralyzed, painful. When you remove the arm in a misguided attempt to get rid of the pain, you get a paralyzed phantom limb you get paralyzed phantom with pain carried over into the phantom. So I said, if I can make the hand move again, maybe he'll feel good. So I said, let's put a mirror there. First of all, I said, let's do virtual reality. And I asked my friends at Caltech, and they said it would cost you $500,000. And this is 15 years ago, and it didn't work very well anyway. So I said, let's take a mirror and put a mirror on this table, and I put the normal hand on the normal right side, reflecting side of the mirror, and the phantom on the left side of the mirror, hidden by the left side of the mirror. Well, it doesn't have to be hidden, it's a phantom, but okay. So you put it on the left side of the mirror, and then you ask him to do mirror symmetric movements, like conducting an orchestra, clapping, waving goodbye, while looking in the mirror. He's going to get a real-time impression that his phantom has been resurrected and is obeying his command. For the first time in 10 years or 15 years. This guy's amputation, my first patient, was 10 years ago. And 10 years he's had this excruciatingly painful phantom. I said, Derek, look inside, move your right hand and pretend you're conducting an orchestra. He said, I know I can move my right hand, but my left hand will not move. I do it every morning to try and get rid of the pain, it won't budge. Do it anyway, I said. Okay, he said, you start. He said, oh my God, you're not going to believe this. It's moved for the first time, the movements in my hand are coming back, flooding back into my mind for the first time in 10 years. And it's wonderful, it feels animated, and the pain, stiffness goes away. I said, close your eyes. Oop. No, my right hand moves, my phantom is stuck and painful. Open your eyes. Oh my God, it's moving again. And so I said, well, maybe this can help patients, and then if you send them home, maybe practice with the mirror. You can get rid of the mirror, and you can unlearn the learned paralysis, and then move his phantom again, and maybe the pain will go away. So we did this experiment. The guy took it home for 2 weeks. Long story there, which I won't go into, but the pain decreased substantially. Then we published a longer paper in Nature saying that this may be potentially beneficial for patients. Many clinical trials have been done. New England Journal of Medicine about 10 years ago, Tsao and company, I think about 21 patients, 7 on the mirror, from very painful, drops in 4 weeks, 1 hour each day, to about noticeable but not very painful pain. You can just experience the pain, but not aversive. You put it on a covered mirror, placebo, or visual imagery placebo. Imagine they're moving the phantom. Pain goes up because they're paying attention to it. Crossover, the pain drops again to near zero, very, very little. Now it's used throughout the world in 1 out of 5 clinics as a treatment for phantom pain. Remember, the other treatments are morphine, painkillers, chordotomy, dorsal rhizotomy. These are all final alternatives after you've tried all this, after you've tried the mirror. Okay, next. We thought we were done with phantom limbs and mirrors, and then we came across a paper by Giacomo Rizzolatti. Mirror neurons, you all know about this. Cells in the brain fire when you move and monkey reaches out and grabs a peanut. The same cell will fire, about 10% of these motor command neurons will fire when I watch Bill here reach out and grab a peanut. So monkey see, monkey do neuron. It's creating an internal virtual reality simulation of the impending action of the person being observed and firing accordingly. Now that's well known. The less well known is the sensory mirror neurons. If you go to the back of the brain, to the parietal region, and you go into the somatosensory cortex, especially S2, about 10% of the cells there will fire, not only when you touch me, but when I watch Bill being touched in the same location. So the cell is firing when I'm touched, or poked, or pulled, And also, if the corresponding limb on Bill is touch, poke, pull, it fires. Now, how the hell does the cell know the difference? So why, if Bill's being poked, why don't I say, ouch, withdraw my hand? That would be a silly thing to do, but why don't I do that? Because I got the neurons wired up for it. Well, I said that's because the receptors in my hand, in my skin of my hand, are telling me, sending a veto signal, null signal, saying, no, you're not being poked, shut up. So it's partially inhibiting the output of the mirror neuron system, enough so that you empathize with Bill, and you say, I'm sad for him, I know what it's like to experience pain in his anterior cingulate or in his thalamus or wherever he's feeling it. But I'm not going to jump up and down and shout, scream pain because my skin is telling me that I'm not being poked. Is that right? Okay, that's clear. Now, what's the prediction from this? The prediction from this, if you remove his arm, if you remove my arm, and you poke Bill, I should feel his pain. And if you stroke him, I should feel his stroke in my phantom. We tested this, believe it or not, 500 years of research in phantom limb, nobody had taken patients with phantom limb, asked him to look at a normal person, stroke a normal person, and ask him what he saw. You do this, and the astonishing thing is, in 3 out of 4 patients, they look at me stroking myself, and they say, yeah, I feel that in my phantom. It's moving, it's moving exactly like the way you move it. You tap it, it's tapping. You poke it, I can feel the hurt. And he says, I said, this is great. We call it, I don't know what we call it. We call it mirror touch synesthesia. Okay, and so the guy goes home and he phones me, and I said, out of just curiosity, I said, you said you get phantom pain now and then. Why don't you have your wife massage your hand? Watch it. Now, I still can't remember whether I suggested it or he thought of it, but one of us thought of it. And then he called me later that evening, and he said, my God, whenever I get the excruciating pain in my phantom hand, the clenching, I simply watch my wife massaging her hand. I get a phantom massage relieving the phantom pain. And I've done it a few times. Now, this we've not tested in a clinical trial. We need to do that. I'm just telling you, it's been published in Archives of Neurology. It's published. We've done several patients, but no clinical trials with placebo have been done, unlike the mirror procedure, which has been done double-blind. So now, to me, that's very important because in a lot of arcane philosophical debates about whether mirror neurons are important or not. I mean, I don't care. I mean, they're helping me make a prediction. I'm saying here's the patient with a mirror neuron signaling that he's being massaged, and that's helping him alleviate the pain. I would have never made that prediction had it not been for Rizzolati's discovery of mirror neurons. Right? So, okay, phantom massage. So the strategy I've been adopting so far, you can see, is to take patients with bizarre neurological syndromes. Some well-known like phantom limbs, but not adequately studied. Some rare like apotemnophilia, xenomelia. And then ask yourself, what can you learn about normal brain function by studying these patients, and can you help the patient? So 2 agendas, clinical and theoretical. This procedure doesn't always work. You only hear the success stories. We see a lot of bizarre patients with symptoms which we don't explain. It's often a wild goose chase. And indeed, there are many established syndromes which I think are a waste of time, or not real. To give you an example, du Clerambault syndrome. How many of you know about du Clerambault syndrome? Raise your hands. Nobody. Okay, well, it's well known among psychiatrists. And this refers to a young woman developing a delusion that an old, rich, famous man is madly in love with her, but is in denial about it. This is an officially described syndrome, by the way. And this is nonsense. I mean, there's no equivalent syndrome described where an elderly, aging, rich, famous man develops the delusion that a young hottie is in love with him and in denial about it. This is very common, by the way. But it's not been described because psychiatrists are men mostly. Second, I'll give you another example. If you think I'm making this, I'll give you another example. Chronic underachievement syndrome is recognized. Used to be called stupidity in my day. Okay. The third one is the best of all, and that is oppositional defiant disorder, ODD. You all know about ODD, right? No, okay. A young, spirited youngster dares to challenge the authority figure, like a parent or a professor or a psychiatrist who made the diagnosis. Now, the man who invented this disease was a genius because any attempt by the patient to protest the diagnosis is further evidence for the diagnosis. Okay? So the neuropsychiatry is full of these examples, and you can waste a lifetime pursuing them. But so if you're lucky, you suddenly see something, you can figure out what's going on in the brain and then help develop treatments. Okay, I thought we were done with mirrors, but we're not. There's this condition called RSD, reflex sympathetic dystrophy, where a patient has any one of you have a tiny metacarpal bone fracture, you get swelling, you get inflammation, you get pain, you get redness, you get warmth, and you get immobilization, reflex immobilization, all the signs of inflammation. Then the bone heals after a few weeks, signs reverse. Warmth goes away, swelling goes away, pain goes away, you move the finger again. Okay, that's fine. In 2% of patients, that doesn't happen. Whether the pain is of central origin or peripheral origin, the pain starts here, finger swells up, is inflamed, painful, immobilized. The injury heals, but the swelling continues, warmth continues. Involves the entire hand, entire arm gets swollen, painful, excruciatingly painful, with allodynia, and immobilized and paralyzed. And the patient is terrified of moving his hand, lest it elicit an excruciating painful sensation. There's no known cure for this. The people go to conferences in the Caribbean about the taxonomy of RSD. Is it type 1, is it type 2, what's the difference between type 1 and type 2, and this goes on, right? No known treatment for it, except things like cordotomy, drugs which are usually ineffective, ganglionectomy, sympathetic ganglionectomy, sometimes partially effective, but usually not. No placebo controls have been done. We said, let's put a mirror there. Because it's known that part of the problem is the guy is terrified to move his hand because it's painful. And there's a Hebbian loop between attempting to move the hand and excruciating pain that then becomes self-amplifying. Put the mirror, have him move the normal hand, look at the mirror reflection, and the dystrophic hand suddenly looks like it's moving, obeying his command, moving around with no pain, right? Because it's not actually moving. And the guy says, oh my God, I'm moving my hand and the pain's going away. And as you watch on the table, now by the way, I suggested this experiment, but I was not formally involved in it, which is a good thing because I'm biased. It was done by a group in Oxford and Bath, and I was reviewing the paper. And on the table, as you watch, the temperature drops. You put in a thermometer. You look in the mirror, look at the reflection, thermometer, temperature drops, swelling subsides, pain subsides, the hand starts moving again. And the guy is in tears. For 9 years, he's had this problem. The guy is in tears. Tested on 9 patients, 3 patients, a few years after the onset of the disease, 3 patients, 6 months after onset, 3 patients, a couple of months after. The first 3 patients recovered completely, the ones who had recent onset. This has now been done in large-scale trials. Okay, this example of RSD. Large-scale trials by another group, and they have done this experiment. The first paper, by the way, was by Peter Halligan, John Marshall, and Patrick Wall. When I said Patrick Wall's paper on a name on a paper, I said on pain. I said this guy is the world's leading expert on pain and on placebo. So if he's got it right, then we know that it's real, the phenomenon, right? So this is a controlled trial on several patients, over 24 patients. Again, you can see, as in phantom limbs, tremendous reduction in pain, down to 0 with a mirror, with imagery and all of that. Nothing happens, pain goes up. You cross over, they're down to 0, okay, down to 1 or 2. So this is now adopted by 1 out of 7 or 8 clinics in this country, in many developed countries. Okay, so that's about, most striking example I know of mind-body medicine, of your mind looking in a mirror, affecting not just pain, but your skin and bones. Right? And this is a powerful example of mind interacting with body. So much for modularity of brain function. Okay, now these are all bizarre syndromes and symptoms. And how much do we have? 5, 10 minutes? I don't see a clock. Oh, okay, good, all right. You can take mirrors and play with them, generate these bizarre illusions in yourself. You don't need neurological patients. And one of the things we did was, I mentioned 2 examples just for fun. If you have hollow mirrors and you stand in the hollow mirrors, and then every alternate image, the ones right next to you are mirror images, and they do what you want them to do. You move your left hand, the mirror moves your right hand. Your brain knows this from years of experience, and you correct for it. Let's not go into the question of why mirrors flip left, right, and not top down. Philosophers have argued about that for centuries, okay? But it flips left, right, so your left hand, the image moves right hand, all is fine. It looks normal like a guy, like you, a duplicate of you. But every alternate image is a doppelganger, which when you move your right hand, he moves his right hand. That looks spooky. And the brain gets an odd sensation. There's something weird going on here. Now, if I touch you and stroke you, you feel the strokes coming from out there instead of here. Because you've not had the compensation, which you normally do for a normal mirror, in a double reflecting mirror, the compensation mechanism goes messed up. So you start feeling it out there. Sometimes transiently you feel you're out there. Most interestingly, if you move your hand, this is relevant to this conference, you move your hand, the first image moves its hand, of course, like any mirror image would. But the second image moves it, follows it, lags it with a tiny time delay. And 9 out of 10 subjects doing this without suggestion experience this lag like an oil. Why would that be? And I'll tell you why. It's a Libé experiment done with a mirror in 10 minutes without any fancy. What's going on is the brain is sending a command to your hand. When you move your normal hand, there's a delay. There's a signal arising in your brain, reaching the hand, moving the hand. Maybe 50 milliseconds, maybe. So the whole process takes 50 milliseconds or so, or even 100 milliseconds. But you don't want the hand to be delaying when you issue a command. So the brain applies a correction and backdates the experience of the moving hand. In the case of a normal mirror, you've used it for a lifetime, you do the same backdating. But when you look at an abnormal illusion in a mirror where the guy's moving the wrong hand in response to your command, this fails to be applied. And the failure to apply leads to a lag. Because there is a lag, which you're not correcting for, and the mirror image lags behind. So you can use the regular mirrors and play with them. Another example is the Pinocchio illusion we discovered years ago. You have a person sitting in front of you. Imagine I'm the subject, experimental subject, blindfolded. And you're the experimenter. You take my hand, my left hand. Is that clear? I'm blindfolded subject. And there's another person standing in between. And you hold my hand, my left hand, and stroke the other person's nose. Right? And what do you feel? You feel, I feel my finger stroking another person's nose, obviously. The person standing here. But now, the experimenter strokes my nose in perfect synchrony. Tap, tap, stroke, 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 tap, 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 like Morse code, tap, tap, tap. In about half a minute, you feel your nose 2 feet long. Because the brain says, what is the likelihood that the stroke and tapping, irregular tapping, stroking of that thing, and my hand is out there, so it must be 2 feet long. But the sensation is coming from my nose. The only solution to the problem is that my nose is 2 feet long. And we've tried it, and you can try it on other body parts too for your ego, but don't quote me on that. Okay, the extraordinary malleability of body image we have discussed. Now I'm going to conclude with one last phenomenon studied by Francis Galton. You may know about our work on synesthesia showing numbers and colors get muddled up in the brain. Numbers seem colored. We figured out that there's cross-wiring in the fusiform gyrus. David Brang, Ed Hubbard, and I, and Boynton figured out that if you go to the fusiform gyrus, there's a color area, V4, and then the number area of Dehane is also right next to it. So there's a cross-wiring. This is the phantom limb cross-wiring. But due to defective pruning of genes, synesthesia genes, which then get expressed selectively in the fusiform gyrus, you get defective pruning, hyperconnectivity, and color sensitivity. But that's another lecture. I won't go into that. I did want to mention the most recent experiment we did, which is a phenomenon called calendar lines, described by Galton. When I ask you to visualize a annual calendar, most of you here will say top left, January, February, March, April, May, kind of does a zigzag, December. It's vague, nebulous sort of thing in front of you. But these people, about 2% of the population, not rare, will see a calendar like a hula hoop. January, February, March, April. It's in Helvetica font. I can see December there written in big letters. I can see the line surrounding the calendar. Now, are they making it up? The same old problem always in neurology or psychiatry for the matter. Are these people just making it up? Or what does that mean? I see a calendar in front of me. An L shape, January, February, March, April, July, August, September, October, November, December. An inflection of the L. Runs in families in a Mendelian fashion. So something is going on here. What? So we then, first of all, do you know it's real? So we said, let's do an experiment. And 100 years people have tried to figure out if the people are making it up or not. So I said, let's do a simple experiment. If I ask any one of you to name the months backwards, alternate months, October, August is the next one backwards, right? -1, -1. It takes ages to do it, and normal people take 40 seconds, 30 seconds to do it. This guy does it equally fast, forward and backward, because he's looking at a calendar. He sees them printed on a sheet of paper in front of him, as though he reads it out, and you can see his watch, you can measure his eye movement surreptitiously, watch his eyes tracking the calendar as he names the numbers. So this guy is constructing a calendar in his mind, but it's floating out in front of him in space. Why and where? Well, we think the angular gyrus in the left hemisphere is involved in sequences. The brain cannot construct. A calendar is very important, by the way, for all of us. You have the sense of being anchored in time and place, your sense of self. You plan for the future. You got a week punctuated by days and hours and months and years. So we all have a sense of calendar in our brain. People usually think it's frontal, but I'm going to argue that the angular gyrus is involved, very much involved. The brain takes things which it cannot compute, like time, and maps it onto spatial maps in the brain, which are everywhere. dozens of them. Going back to the Devonian period, you've got spatial maps everywhere. Map time onto space, map the calendar onto space. So that's why you get a calendar here, and then there are fibers going from the angular gyrus, and you can show this as a DT imaging to the hippocampus, with the place cells and the grid cells, the GPS of the brain, the inferior longitudinal fasciculus, angular gyrus, and the place and grid cells together constitute the human calendar, aspect of your self-awareness. Thank you.
Thank you so much. We have time for a few questions. Well, let me ask while we're, do you ever get jealous of some of these people? Would it be cool to have a calendar line and sort of mental capabilities? I mean, you've had so many interviews with interesting people.
Yeah, well, one does. I mean, it's a unique experience, and certainly synesthesia, where people experience music and colors and see the numbers, see a richer world.
Yes.
And that I have been envious of, but not opportunophilia or
Yeah,
kind
of course. Of course. Thanks for running, Betsy.
Thank you. Just the other week, I shared with my dad the new VR headset that Samsung had, and both of us had quite a remarkable experience with that. Just hearing the experiment with the mirror, I'm wondering if anyone has suggested trying to alleviate phantom pain with using VR and virtual reality.
Yeah, it's a good question. And in fact, when we started out, we thought of using virtual reality first, and then it was too expensive. So we used a $2 mirror. and then later on, people started applying for grants using VR, and then we wanted to apply, but then you can't ask for a lot of money for mirrors. but with VR, but jokes aside, you can use it for bilateral amputees. You have to use VR. You can't use a mirror. So but there's a very small minority of amputees are bilateral. I don't know if that answers your question, because the first part of it was a bit, I couldn't
It's
get.
possible. It would be possible to do it with virtual reality.
Oh yeah, yeah,
Sure.
it's entirely possible. Yeah.
Sure. Down here. Sure. Let's take that one. And then we have one down in the back there. Yeah.
So
Go ahead.
similar to your calendar and counting back through the months, has there been any work done on number lines and counting up and down, whether people count faster or slower, depending whether their number line goes from bottom to top or left to right?
So are there any other kinds of representations, such as a vertical number line, that people have a visualization of, or analogous things?
Very good question. People do have number lines that they have been studied more than calendar line. There are only 2 or 3 groups who have studied it, and we've just started studying it. But number lines have been studied for a long time, including in normal people using reaction time paradigms.
But they can see it in their mind's eye.
Yeah, there are some people with a number line which is twisted. Galton said, instead of saying left to right, with a normal number line, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, with your Arabic, I should say Indian numbers, one to left to right, they have a convoluted something that does, and they'll draw it for you. So that doubles back on itself, and 75 may be actually closer to 2 than 5 is closer to 2. And this runs in families, and people have established that it's almost certainly a real phenomenon.
So is there a mapotopic number line that corresponds neuroanatomically, like
Well,
the
nobody's
homunculus?
looked for it, and I think it's very likely that, again, the same thing is going on. You're mapping numbers, numerosity, ordinality, cardinality onto space, like on a graph paper.
Sure.
And this gets messed up some way in some people, and then you get these odd representation of numbers.
Yes.
And people have used the various reaction time paradigms to study that. Good question.
Very good. This one down here.
All right. Thank you for a fascinating talk. So hear about the phantom limb phenomena with amputees, but I'm wondering, do you ever see this with people with high spinal cord lesions? And if not, why not?
Okay, with spinal cord lesions, can you see phantoms? The answer is absolutely yes. If you have complete paraplegia instead of partial, then the patient, if the eyes are open, he sees his legs there, so by definition it's not a phantom, right? But if he closes his eyes and goes to sleep, next morning it may be occupying a strange position. And he wakes up, does a double sip with his eyes, and he says, oh my God, my phantom is, my legs are over here, not there. And so you get that kind of phenomenon where you certainly get very vivid phantoms, sometimes painful phantoms. And people are using VR to treat, partly inspired by or following our work from the mirrors in paraplegics, showing substantial recovery of function.
One last quick question.
Not function, sorry, from pain. Yeah.
I'm curious if you've got these, all this work in mirror neurons and their ability to, to kind of create this signal inside one person's mind about someone else's experience. What does it say about the nature of our minds that, does it, what kind of implication does that have for how widespread our minds are, to the extent that they can extend into other people's minds?
I'm sorry, we're still having a little trouble hearing that. Can you try and speak up a bit?
What role do mirror neurons play in extending the mind out to others?
Oh, okay. What role do mirror neurons play in extending your mind out there to others? Yeah. Well, almost by definition, they're a form of shared consciousness, as somebody was saying yesterday. But you get into trouble saying things like that. I mean, I find that you talk about mirror neurons and start saying it dissolves barriers between people, and people get riled up. So I avoid saying that. I mean, you just present the empirical results, and you find that this is what happens. You massage somebody, you feel it in your phantom limb. To me, there's a dissolution of the qualia barrier. You're literally experiencing somebody else's qualia, which would ordinarily be considered impossible. Now, things like empathy, too, are probably mediated by mirror neurons. The ability to take somebody else's point of view in judging a situation may require participation of not the sensory mirror neurons, but some other class of mirror neurons. There are mirror neurons for emotional expression, too, in the amygdala, yes, and insular cortex as well, especially in insula. So, yeah, so, I mean...
Thank you so much, Dr. Ramachandran. Well, I was just being asked why someone would want to be in San Diego, especially if somebody was interested in neuroscience and cosmology things. And I'm hoping we're demonstrating that a little bit this afternoon, as well as with our fantastic talks yesterday. So carrying on with our sister institution to UCSD, we have Dr. Charles F. Stevens from the Salk Institute and at UCSD talking to us about the evolution of brain mechanisms for consciousness. Dr. Stevens.
Thank you. I'm very pleased to be here. As far as I know, we have no idea what the brain mechanisms for consciousness are. And so I'm going to assume that the brain mechanisms for consciousness are not going to be that different from all the other brain mechanisms. So I'm going to talk about what brain mechanisms would consciousness might be like if we knew which ones there were. Presumably, there is selection pressure, and consciousness is something that improves our chances for survival, and so they would be selected like everything else. Now, the first point I want to make is that all brains are different. The smallest one here, okay, this is a mouse brain. This is a monkey brain, and this is a human brain, and it's absolutely obvious that they're all different. One is that the mouse is much smaller, the human is much bigger, the monkey's in between. They also have a different shape, each of them, so they're clearly not the same. At the same time, you may not know that the human brains by themselves are all different. So if I were to count the number of neurons in the visual cortex of everybody in the room, what we would find is that some of you have 3 times more neurons in your visual cortex than others. There's somebody in this room that has fewer neurons in their visual cortex than a normal chimpanzee. In spite of this, we can all see well, and it's not just the visual cortex that varies in size between individuals. It varies in size within one individual between the left side and the right side, and all the other areas also vary in size. And this is partly but not entirely inherited. And so all of our brains are different, even the brains of people who are identical twins. At the same time, all brains are the same in the sense that I'm going to tell you. V1 right here stands for the primary visual cortical area. It's at the back of your head here. And V1 is going to be used as an example a number of times through the talk. So I'm going to be saying primary visual cortex or V1, but it's all going to be the same. And all mammals, essentially all mammals have a V1, and it's shown here in colors. It's the red color for the mouse. There's a cat brain there. It's the red color for the cat. And the human, it's the red color for the human. The human brain, the human V1 is actually much bigger than it looks like here because it folds around on the inside. Also, all the parts of the brain are the same in all animals, the same way that parts of the body are the same. You all have a heart, you all have a liver, you all have intestines. They're basically the same from one person to another. There are important differences, but they're the same. And and different parts of the brain are the same. Here, one of the parts of the brain, which is the cerebellum, is here, and it's here, and it's here, and all animals have a cerebellum. Not only that, but if you look at each one of these areas, like the primary visual cortex, they all basically work the same. You can study a mouse primary visual cortex and learn basically how a human visual cortex works. You can study a monkey visual cortex. You can learn basically how a human or a mouse visual cortex works. They all work the same way. They all have the same circuits. They're all based on the same circuit. In the cerebellum, there are only two major cell types sitting in the cerebellum. One is called Purkinje cells. Those are the bigger cells. And the other one is called granule cells. Those are small cells. About half of the neurons in your brain are granule cells. They're tiny and they're gigantic numbers of them. Not only are the cells the same, but they're related to one another. And this is a graph here where I've counted the number of granule cells, and these numbers are in millions. And so the humans are up here. And there are a bunch of different points from different studies for the human cerebellum. And you can see that they have a little bit more, maybe 200,000 times a million neurons up there. So it's a lot of cells. And there are about a little bit less than 100 million Purkinje cells, which are over here. So they're a gigantic number of cells, but the cells at this end are from the mouse, and those cells are there are many fewer numbers. There's still an awful lot of them. And in between the humans and the mouse, there are a bunch of other species that are here, and you can see they all fall on this line on a double logarithmic scale, and the line doesn't have a slope of 1, it has a slope of 2/3, which means that the number of Purkinje cells is proportional to the number of granule cells to the 2/3 power. It's not a linear relationship, it's a power law relationship. And this is what's called an allometric relation. If you have things that are scale linearly, it's called an isometric relation. And an allometric relation is one that does not scale linearly, but scales on a log-log plot by a straight line. This was originally coined by Julian Huxley, who is Aldous Huxley, the author's brother, and he was one of the people. He's the person who sort of started the study of relationships between animals that are scaling like this, and there's a deep meaning for these allometric relationships. The meaning is that the parts of the body are sometimes bigger and sometimes smaller, and there are a bunch of ants over here. And these are just drawings of ants, but you could know, and these are all the same species, but some of them are worker ants, and some of them are army ants, and you can see that the biggest ant has a head that is much longer relative to his body than the other ants do, but it's the same shape. And what I did here is I just traced all of the outlines of all the ants and used Photoshop to squish them to the same size, and you can see they all have essentially the same shape. And this is a normal thing, but at the bottom here is Darwin's finches. And these are different species of finches from the Galapagos Islands. And each one of these species has a different shaped beak. There's a very large beak for this species and a very small beak for this species, and other beaks are in between. And they're all the same shape, but their sizes are changing relative to the size of the head, just like the ants. And there is an allometric relationship between those two. Where do these allometric relationships come from? They come from the genetic networks that are used for generating patterns during development. And evolution normally works on these genetic patterns. It's known in the case of Darwin finches exactly what you have to change for going from one finch to the other, and you can change a certain thing in the biochemistry of one finch and make its beak change shape the same way. And this is very evolvable. It evolves very quickly. The big beaks are used for crunching seeds and things. The small beaks are for reaching inside little cracks and getting insects to eat. So so what evolution does then is it uses the same pattern formation mechanisms, and it mutates them sometimes. It changes them around, but usually they stay with the same shape. So everything is related to everything else. So the reason that all of the different parts of the brain are pretty much the same is because they evolve from one another. So evolution doesn't start from scratch ever. It always starts from what it has and it changes it. And a lot of times what it changes is it just changes the size of beaks like the finches are. It doesn't change the pattern formation mechanism. It just changes the size of components in it in a way. And the same is true for the nervous system. So basically, all nervous systems are related and all work on essentially the same mechanisms. And this is why a lot of people believe that, all mammals, or maybe all animals, including insects, are conscious, because if one is, then they figure maybe the other one should be, because they're sharing brain mechanisms. But not all animals are the same, and in fact, primates are different from everybody else. A lot of animals are different from everybody else too, but the primates are in ways we know, are different from all the other animals. And one of the ways has to do with their visual system. If you look at different areas of the brain, and you count the number of neurons underneath 1 square millimeter of the cortex, how many are there? Across different species, and the species that were studied here are mouse, rat, cat, monkey, and also humans, and some other animals. Those are the ones that are on this graph. And the figure down here, this figure, that's funny. Okay, I have to plug this in again. Okay, underneath a square millimeter of cortex across all animals, including you, there are 100,000 neurons. Sometimes the cortex is thick, sometimes it's thin, and the figure in the bottom left is all of the data in the literature on these species from a dozen different laboratories that was all collected together. And you can see that they all, on this bottom line here, cluster around about 100,000 neurons underneath a square millimeter. So they're all the same. There is a difference, however, and that difference is in primate V1s, any primates, there are 2.5 times more neurons underneath a square millimeter than in all other species with vision that have 100,000, like cats have 100,000 in their V1. Dogs have 100,000 neurons under every square millimeter. Primates have 250,000. And so, as a result of this came about by mutations when the first primates were evolving many years ago, and there were 2 main things that were changed. One was that primates got better color vision. The second thing was that they had 2.5 times more neurons in their V1 than all the other animals did. And the consequence of that is that they have better vision, better acuity than all the other species. So on the graph in the upper right-hand corner is the visual acuity measured on a number of animals, and the area of V1 in those in animals is drawn. And the bottom line is different species, including some marsupials and things like that, but mainly mammals. And they fall on one line. As you get a bigger visual cortex, you have better acuity. But up above are a bunch of primates, and those primates have, these are log scales, and so they have somewhere between 3 and 10 times more better acuity than any other species we do. This is probably why we're here, because in the early days when primates were first evolving, they were little animals climbing up in trees. And they could see the food better than everybody else, and they could tell the quality of the food better than everybody else because they had better acuity, and so this gave us an advantage, which is a very significant one, and it may be the reason why we're here now, because we have and still have an advantage over all of the other animals, even animals that, hunting animals, for example, a panther or something like that, you would think would have extremely good acuity, and they have better acuity than other animals because they have a bigger primary visual cortex, but they all have less good acuity by 5-fold than we do. Okay, so primates can be special, and they're special in another way. That other way is that we are the only animals that have really good language. Other animals communicate with sounds, for sure. They have alarm calls and things like that. But the other animals, no other animal, even animals that have been taught to speak, like parrots, have language that is anywhere near as rich as our language. And they can't do the kinds of abstract symbolic manipulations that we can have, and so this has given us another advantage: the fact that we can think about things using language that other species can't think about if they in the same way if they don't have language. So. If I had to guess, I don't know, but if I had to guess, I would guess that consciousness, or whatever the neural mechanisms that underlie it are, is not unique to us, but it may be different for us than, say, a monkey, and it might be different from us for, say, a dog. So Christoph Koch, who was at the second one of these meetings many years ago, once told me that he was absolutely sure his dog was conscious. And I thought about that, and I tried to imagine if I were a dog, what my consciousness would be like, and I failed. I have no idea how consciousness would be if I didn't have language. Now, not everything we do is conscious. There is a field of psychology that's been well studied, which is called learning about without awareness. And you can learn behavior, but you don't know what the behavior is that you've learned. You have no idea what the behavior is, what it is you learned. You can do it, but you don't know what it is. And we can have thought without consciousness too, and pretty deep thought. So all of the people, this is something I've never read a paper about. I don't even know if there is a paper, but a lot of my friends are physicists, and this is an experience I've had, and it's an experience that every one of them has had, and it's the following. If you're working on a problem, a math problem, and you can't solve it. You don't know how. You think about it for a while in the afternoon. You can't figure out how to solve this problem. Doing an integral, any sort of problem. You go to bed at night, and you wake up the next morning, and instantly you know the answer. You know how to do it. This happens to all the people who do math all the time. They even know it. Sometimes you don't even have to sleep. You just have to go for a walk. You go walk around, and you come back and you know the answer. And a lot of times, if you're working on a problem, and I'm sure many of you have had this experience, you're thinking about a problem and you don't know what the solution is, and then all of a sudden it comes to you out of the blue. You have no idea where it came from. You were not conscious of the process that got you that idea, but it turns out you're conscious once you see the idea. You say, oh, I bet that'll work, and you try it, and it does. So we have thought. We can think in pictures. We can manipulate pictures, and I don't know if this is true, but my guess would be there are a lot of animals that can use tools. And they don't learn using the tools necessarily from watching other animals use the tools. They figure it out. They just figure out a way of doing it. And what they must do is they must think about the problem they have in some way, and then see a stick and recognize they could use that stick to solve that problem. And this is a kind of sophisticated thought. But it doesn't necessarily involve, that kind of thought doesn't necessarily involve consciousness. And in fact, much of the thought that we have is unconscious. It just comes along. I have a friend who thinks about problems, and what she says is that she works on the problem by thinking about it in a way she can't describe and I don't understand. But then after she has figured out her problem, she translates it into language so she can explain it to other people. And I haven't taken a poll of people, but there must be other people who are the same. I don't know. But I do know that everybody who does math has this experience of not being able to solve a math problem, going to sleep or taking a walk, coming back, and you're not even thinking about it, the problem consciously while you're doing it, but you know the answer. Okay, so I couldn't picture what if I were a dog, even imagining I could have vision, I could have images, and I could maybe manipulate the images. I still couldn't imagine what it would be like to be conscious without having language. And this is probably just because I've had language most of my life, and I use it all the time, as we all do. And so, if I hurt myself, first thing I think of is well, that was very dumb. Why did you do that? You should have known better than that. And then I think, oh, that's really hurting a lot. Is the pain going to get better or worse? Should I go to the doctor? Maybe it needs an x-ray. Is it going to go on for very long? Is it going to get worse before it gets better? Is it something I should really worry about? It's a serious thing. And my experience of hurting myself, it involves me talking to myself about my experience. And I'm not saying that nobody can, but I certainly can't imagine what it would be like to not use my language for thinking. And so I'm suspicious that Christoph's dog, even if he is conscious, he can't be conscious the same way that we are, because I think probably most of you are like me. I don't know. I've never asked anybody, and you can tell me if it's wrong. I think most of you would have a hard time experiencing things that you're conscious of without language as part of the experience. Okay, so I know for sure that I'm conscious. I think you're probably conscious, although I can't be positive, because you act like you are. And if I talk to you about it, you talk to me about the way somebody who's conscious would. But that isn't really such a good test, because these days Google could make an artificial neural network with deep learning that would allow the network to answer any question and discuss consciousness with you as well as any one of you can with each other or with me. And I don't believe that the artificial neural network is conscious, because I know it was trained and I know how it was done, and I don't see any way that consciousness came into that. And so you can't really know. Maybe some of you aren't conscious, but you're embarrassed, and so you won't admit it because you feel like everybody should have consciousness. I don't know if that's true or not. At any rate, if consciousness does come from the brain, I believe that it must have evolved. It must give us an advantage because we have a theory of mind and we can put ourselves in other people's places and predict their behavior better. And I think that may be part of what being conscious is and partly how it evolved. I believe that other animals must have the same neural machinery that we are using for consciousness, maybe not the same way, but I would expect that they would have that they would do things pretty much the way we do, maybe not as well. Their language is not as good. Their being able to manipulate ideas is not as good, and so they may not be able to do it as well. But I can imagine they might be. I can't imagine a fruit fly as conscious. They seem to me like a little automaton. But since I don't have a test for consciousness, I can't really know. At any rate, I've tried to give you a perspective about consciousness based on a neurobiologist's view of how the brain works, and I'll be interested to see how you react to this. Please. Now it's working.
Good, of course. Thank you, Dr. Stevens. Some questions? We'll take the first one over here.
Thanks so much. That was terrific. Yeah, all these years I thought a hawk could see better than I can, like way up there in the sky and then come down after a little tiny mouse. But it's reassuring to know we have that acuity. What about the other senses, like hearing and taste? Are we at the top?
Oh, Dr. Stevens, speak at the microphone. There you go.
Yeah. Sorry. Hearing and taste and everything else is the same as in all the animals. Their specializations, for example, bats use their squeaking and their hearing for echolocation, and they have special cells that are used for this. Cats can't taste sweet because they don't have any sweet receptors, so they're not interested in that. But there's some special things like that. But in general, all the species are the same for vision, for motion. All the things works essentially the same way in all species, although when it's demanded of you, then you have specializations of it.
No, I see that. But wait, I thought you said that acuity, visual acuity, primates were superior.
Visual acuity in primates is superior. Is that not correct?
Yes, it's 3 to 10 times better for the same brain size in primates than it is in any other species. But that's true across all the primates, not just us.
Okay. Next question here.
Yes, this is a question about one of the things you said is, I'm here.
Oh, there you are. Yeah,
are you aware?
They have us blinded up here.
The purpose of yoga is to give you the experience of consciousness without language, and the ability to have ideas primary to language. When you look at the origins of yoga, they talk about 4 levels of speech. There's the speech that I'm speaking, there's the words that I'm thinking before they come out, then there's the level of ideas, which underlies that. I'm actually presenting a model of thinking which can incorporate putting the ideas before the language. But when you look at a sheepdog trial, for example, the shepherd will give whistles in different frequencies, and those whistles will transmit the idea to the dog, whether to go right or to go left, to go further or to come back. And the ideas get transmitted without the use of words, but with certain kinds of cue. And this has been also seen in pets and other things. There's direct communication. Are you aware of any of those things? Because they seem to me to be very revealing.
Yeah. So the people who have had strokes in the right place can think absolutely perfectly, but they can't often say the words, but they can write them down. So they know what the word is, but they can't think of how to say the word. They can write the word down and get it. And when they come to one, they can't figure out what word to say. They can do that. And so I've never asked anyone like that how they do their thinking, but I know they do because I've had friends with strokes. One was a physicist, and he still did physics. Still could do mathematics, so he definitely thought some way, but I don't know how he thought or how he or what his consciousness would like. Be interesting to see.
Yeah. So how can I phrase this as a question? So I am one of these people who thinks visually, and in fact, if you watch me walk around the conference every now and again, because it's so intense, I will have to stop completely still because I'm using my visual cortex to think, I guess. So the question is, can you test me? Is it the fact that I'm dyslexic and I'm compensating by using my visual cortex, or is it just a different way of thinking?
Did you get that?
I didn't.
Okay, so he's suggesting that when he's deep in thought, he has to stop, and he may be retasking his visual cortex for thinking purposes. Would there be any way of testing that and examining that?
Yes. Okay. There are people who are blind from birth, and very frequently they are very adept at music. If they became blind early enough, very many of them have perfect pitch. And are prodigies at being able to perform music. But people like that, you adapt their visual cortex for doing other things. And so there have been studies where they've looked at people who had been blind for most of their lives and have given them tests showing that they can think about things with their visual cortex, using their visual cortex. They can do an MRI and show that they're using the cortex, and they can do it better than, say, I could. And so if there are areas that can be taken over and can be used for thinking, for sure, and visual cortex is one of them.
Two more quick questions here.
Yes, I would like to share with you this question, consciousness without language, from a non-verbal autistic point of view. Working with them, some of them can type, but not speak. They say that their thought and their perception of reality don't fit in the poor tools of words. They have a highest consciousness from their point of view. And they also say that without words, they perceive instantaneously the dynamic interconnection of everything. And from their point of view, there is, in our language, like a breakdown of correlation between the reality, the perception of the reality, and how we describe it by words. And so that's why they get nervous, because they can't fit in the words the biggest things of reality that they perceive. Sorry for my English.
You did fine.
Can people like that read a book, for example?
Can they?
Yes, but they can't.
Yeah, I can understand that. But if they can read the book and understand it, I can imagine they could think in a kind of in a language kind of way, even though they can't actually speak. I can imagine that. Yeah, I don't know.
All right, two more quick ones. Very quick.
I wanted to just put in again a plea for consideration of the glial cells, because as far as I know, there is a non-allometric relationship between the number of glia and neurons in certain parts of human brain compared to even primates, including, most importantly, frontal lobe. And that may feed into how our brains are significantly different in creating the processing complexity. What do you think?
Well, I don't know. So in Los Angeles, they've heard of glial cells. Have they heard of glial cells at the Salk Institute?
So the number of neurons and the number of glia are different in different sized brains. But what matters is the number of synapses that there are. If you know the number of synapses that are in the brain, then you know the number of glia, because these are astrocytes, because they cover the synapses. They make they embed the synapses, and so there are studies where they show that if you that even though there's not a good relationship between number of neurons and number of glia, that there is a very tight relationship between the thickness of the cortex and which is basically how many synapses there are, and the number of glia that are there.
Last quick question.
Hi. You said that it should be advantage evolutionary. I'm over here.
Okay, your sound is over there, so that's why I keep looking over that way.
Yeah. Excellent presentation, by the way. But you said it should be an advantage, evolutionary advantage. But on the other hand, if we can think complex thoughts and behaviors without consciousness, then what should be the advantage of consciousness?
What's the benefit of consciousness if we can have thoughts without them?
I think that I mean, I don't know what the advantage of consciousness is. I'm glad I have it, but I don't know. But I would guess, and that is that if I can think in the way that you do, I can put myself in your place, and if I know you well, I can predict your behavior better than without it. And most people, when they're thinking about something, they can think without being conscious necessarily. But most people, when they're deciding what to do, they talk to themselves. I haven't taken a poll. I don't know if there are a lot of people who don't talk to themselves when they're making a plan for what they're going to do for the day or something like that. But I bet most people are that way. They do it the same way that I do it, which is I think, okay, now what do I have to do today? Here's a list of things. I make the list in my head, and so forth. So I would think that this would give me a better theory of mind, so that I could predict other people's behavior better. I would think that would be the advantage.
And with that thought, we'll finish this afternoon session. So just